Here I'd like to show you some of the examples in plants for the plant HGT, and well, because I'm more familiar with plants, I think that's the major reason. I could use the fungi example as well. But anyway, so as we can see here, in plant HGT cases, we know most of them involve mitochondria. So very few HGT events happen in chloroplast. So that's due to some reasons in the property of mitochondria in plants, because in mitochondria genome in plants is very dynamic. It's very there are very high frequency of recombination and and rearrangement. So there's probably a reason why it facilitates the HGT event. And there are some evidence I can show you from some of the old literature to show you how those events were discovered, including the COX-1 intron and RPS2 and NAD1 intron and ATP1 in parasitic plant. And there's a very special case in Borrelia and Needham, and we'll talk about it later. So first we talk about the mitochondria COX-1 group 1 intron in angiosperm. And mitochondria genome in animals are very conserved in a way. I mean the gene arrangement. But in plants, this is kind of very difficult to study. And in the COX-1 group 1 intron, it's usually it's not present in angiosperm. It shouldn't be there, and it's present in other organisms. But in 1998, Keith Adams found there's a piece of intron found in peperomia. It's a kind of paper based on the southern hybridization. And so this is the first case, and originally they couldn't figure out what's going on, and eventually there are some follow-up studies and survey a lot of angiosperm genera, and they found 48 genera contains this COX-1 group 1 intron. And this is the original data from southern blot, and these are the different plant species. So they use this COX-2 group 2 intron as a control. So every species are hybridized, so the DNA is there. And they also so they use the COX-1 exon and COX-1 intron as a different probe. So COX-1 is present in all of the plant species here, but the intron only present in some particular cases. And this is just one part of it. They study quite a lot of different plants, and eventually they map all the 300 genera within the angiosperm, and found out that there are many cases for the presence of the COX-1 intron. And those ones with the very bold face or thicker line, and all the things with some color on the taxon name, those ones show the presence of COX-1 intron. So there are 30 clades of this intron-containing clade were identified. So the next question will be, how do we know it's a horizontal gene transfer, or it's a massive losses for among the different lineages? So the very easy way is to compare the phylogeny. That's the most easy way. So we have three different trees here. The tree on the left is the COX-1 exon plus intron tree. So this is kind of a mix-up for that, and it's kind of hard to understand on this point. But if we compare those two, they'll be more meaningful. And for the tree from RBCL and COX-1 exon, and you will get a tree something like this one. So this is considered to be the species tree, which are also supported by the 18S ribosomal RNA sequences. So we basically use this grouping as the color code. So the orange, yellow, light green, blue, and purple for large groups of the particular plants. And we map the color on the tree derived from COX1 introns. And we'll see the tree from the COX1 intron does not support many of the relationship for the true species tree. And in this case, you will see the orange thing does not group together. The yellow things are here, and so on. So basically, this gives you some idea that there's incongruency between the gene tree and the species tree. So the best explanation is that there's something happen between those distantly related species, that there's some transfer. The piece of gene sequences was transferred between those distantly related taxa. That's the easiest way. Because there are two scenarios you can imagine. If this spatial piece of DNA, the COX1 intron, was transferred or from non-plane to plane, then the phylogeny will probably look like this one. That's one possibility. And if there's a single event from the non-plane to plane, and then there's some transfer between the plane, then if there's only single origin for this COX1 intron, then the phylogeny should something look like this one. And based on the phylogeny we just saw from last slide, it should be the upper one. So it means that, so basically we try to reconstruct history. For example, well, if you imagine this piece of intron was from maybe a virus, or as a vector, or from somewhere, and if it only happened once, then it should be and then transmitted either between using the grafting or hybridization or whatever, then the phylogeny will be very similar to D. But if it's multiple hits on several different clay, then it should be something like the B. So what does that mean for the multiple hits from the donor? And there will be something, this piece of DNA was carrying around by several different things, and it was attacking something here, attacking something there, and then it's very easy to transmit it here and there. And then we can find a very special case like this one. So of course, the next question would be, what is the mechanism? How does this happen? And of course, there's no easy answer for that. And we have little idea. And this this intron does not have the normal mobilities like the group 2 intron. So it's not very easy to cut themselves and then to transfer to other part of genome. This is not as easy as the group 2 intron. So at this moment, we still have very little idea how this happened in this particular group.